Hi, it's great to be here. Vik Khurana from Brigham and Women's Hospital and Harvard Medical School. It's always a pleasure to give this particular research update every year. Today, I'm going to be joined by my colleague, Dr. Diego Rodriguez. Diego is a physician and is a clinical research fellow with us with a special interest in multiple system atrophy. Welcome, Diego. Today, we're going to together do something a little bit different. In science, when we're interested in a particular paper, we journal club the paper, we take a deep dive, we talk about its context and its implications. And in a way, that's what we'll do today. We'll take one interesting paper, an exciting paper from 2023, and we will journal club that for you, give an appropriate introduction, explain the context and how we're using findings from papers like this in our own research. So we hope you think this is interesting. I'm going to share my screen now if I can. Hopefully you can see that. And let's start the slideshow. And let's get going. So the paper that we're going to cover is this one. It was published a few months from the group of the Hattori group in Japan. It was published in Nature Medicine. And it really involves the diagnostics of synucleinopathies, diseases associated with the aggregation of alpha-synuclein. And as many of you will know, multiple system atrophy is one of those. So this is a paper that has direct relevance to all of you. Now I'm gonna introduce the context here a little bit, take you through why this paper is important. And then Dr. Rodriguez will discuss the paper, and then we'll close out with a few ways in which this paper is helping our own research. So this is a throwback to earlier presentations where we discussed why the coalition is working on specific research projects. It's really to answer some of the fundamental unknown questions in the field. And I'm going to recap this. These questions were crystallized for all of us in the community when we met together as an international community in Las Vegas in 2014, developed something called the Global MSA Research Roadmap. So the first question is what causes MSA? We don't know that. There are genetic causes, potentially environmental causes, and the coalition and other groups around the world are very interested in answering this question. Where does the disease start? That's number two. That's another way of saying where is the inciting pathology? Where does the disease process begin? In which cell type of the brain? And if we were to model it in the lab, how would we do that? Another very important question. What drives progression and how do we measure it? We need biomarkers in this field. We need to be able to diagnose this disease early and to be able to understand in a relatively short period of time whether some intervention like a clinical trial drug is actually slowing the disease down. So for that, biomarkers are important, and you don't get biomarkers unless you have biospecimens. And so this is point three, which is fundamentally important to the early diagnosis and developing appropriate clinical trials for patients. And of course, the big one, number four, is how do we treat the disease? Until now, multiple system atrophy patients have had symptomatic treatments that can help them with day-to-day living, and there's many things that we can do, as you know, and many good sessions in this conference on that. But what we're all, of course, hoping for is an ability to prevent this disease, reverse this disease, at least slow it down. And amazingly, we're seeing in different areas, ALS, Friedreich's ataxia, Alzheimer's disease, we're seeing FDA approvals coming through now. So as a field, we are tremendously optimistic that neurodegenerative diseases that for decades have presented such a challenge for us as patients, caregivers, and clinicians, that those are becoming a tractable problem to solve with research. So the paper that we're going to talk about really fits into points 3 and 4. The paper describes an interesting new biomarker in the blood, and that has implications for a set of treatments that are being developed for MSA. Many of you will know quite a lot about MSA. Some of you will be newer to the field. I'm going to give a little introduction to the disease and its pathology, and I'm going to focus on points that really are relevant to the paper that we're going to discuss. This is from a very nice review from Gregor Venning and colleagues, in which multiple system atrophy is described in some detail. It's a rare disease, as you know. It comprises of autonomic dysfunction, bowel, bladder, blood pressure, sexual function, and so forth, in combination with a motor disorder that can cause features of slowness and rigidity that are akin to Parkinson's disease, and hence MSAP for Parkinsonism, or can lead to a different beginning, and that is a motor disorder that results from staggering and imbalance, and that would be the MSA cerebellar type, MSA-C. The clinical course is depicted in the figure from the review. You can see that on the lower left of your screen. The autonomic issues in the left, sleep disorders. Many of you will know that enacting your dreams is a fundamental part of this disease for many patients, and then progressively the motor disability, and you can see it progress, unfortunately, from imbalance to a wheelchair, and increasing disability over a decade or so. So this is the natural history of the disease that we're really trying to ameliorate. And the idea behind the paper today is that if we could identify MSA in early patients with a precise diagnosis, we would have so much more ability to get the patients the treatments they need early in the course of the disease and actually make a difference. This is considered to be a very important aim of any disease-modifying therapy of a neurodegenerative condition, is really implementing that therapy early. But you can't do that unless you make a precise diagnosis. And many of you will know that one of the most frustrating aspects of the MSA journey is at the beginning, often bouncing from doctor to doctor, neurologist to neurologist without a clear diagnosis. So the early diagnosis is important. And then once you start some kind of treatment, these days it would be an experimental treatment because we have nothing out there in clinical use right now that slows this disease down. But for a clinical trial, you will want to know if your treatment is doing something beneficial to the patient, and you don't want to wait years to have that answer. So it's coming back to that original question of biomarkers. This is a depiction of the pathology here, just showing the different distributions of pathology in the cerebellar type and the Parkinsonian type in the brain when we see them. Now, a really key part of this paper that we're going to discuss relates to alpha-synuclein. So alpha-synuclein, as some of you will no doubt know, is a protein that's expressed at very high abundance, predominantly in neurons in the brain. And in a group of diseases called synucleinopathies that include Parkinson's disease, multiple system atrophy, and dementia with Lewy bodies, this protein is very conspicuous in the brain. Now, in multiple system atrophy, the distribution of this protein is different to Parkinson's disease. What you're seeing here is you're seeing that there are neurons. Here you can see a neuronal fiber, but each of these neurons is ensheathed with insulation that's coming from a different cell in the brain called the oligodendrocyte, which is the insulating cell of the brain. And it is in these cells, so-called glial cells, that's what the G stands for here in these acronyms on the right, that you start to see alpha-synuclein. And what you're seeing is almost these crescent-shaped structures in these oligodendrocytes. This is the characteristic pathology of MSA. The protein alpha-synuclein should not be mislocalizing and accumulating like this. And because one oligodendrocyte, one of these ensheathing cells, takes care of many neurons, you can imagine when they start to get sick, many neurons succumb. So this is a real problem. These are inclusions within neurons themselves that you can also see in multiple system atrophy. So very diverse pathologies, but a characteristic feature is the accumulation of this protein. Now, just like when you hurt your hand and you cut yourself, you all know that you get pain in that local area and you get inflammation where the immune cells come in to try and heal the damage. Something similar happens in the brain, but the players are a little bit different. And in the brain, there's a type of cell called a microglial cell and another one called an astrocyte. And these are key cells that are involved in neuroinflammation and the reaction of the brain to an injury such as that occurs in multiple system atrophy. By injury, here I don't mean a traumatic injury, I mean a pathology within these cells also induces an inflammatory response. And that's what is meant here by reactive microglia and reactive astrocytes. They're reacting to the pathology. So different types of treatments will be targeting alpha-synuclein. Other types of treatments will be targeting inflammation. And this is just another example of the very different types of responses in the brain between MSA on the left and Parkinson's disease on the right. So while these diseases are related fundamentally through this protein alpha-synuclein that accumulates in both of them, the pathologies, where you see the pathologies and which cells are reacting and how they're reacting is actually quite different. This is a striking image of the cerebellum, part of the brain, back of the brain, that is very involved in balance. It is affected initially in MSA cerebellar type. And here you can see a normal brain with this blue staining, which shows these myelinated fibers, these insulated fibers that I just talked to you about that are generated by these support cells called the oligodendrocyte. Now you can see very strikingly on the right side that myelin has disappeared because those oligodendrocytes are dying and the myelin is simply not found in the brain of this patient, and therefore the signals between the neurons cannot travel as quickly because the insulation is lost. Okay, so next up, just a little summary that includes some jargon that you may hear. And so this is essentially the key elements of what I just told you. There's a neuron here, there's an oligodendrocyte that's causing the myelination, insulation of these neurons, so the signal can jump very quickly down. But you can see this oligodendrocyte is accumulating alpha-synuclein and the myelin is therefore breaking down. And so this alpha-synuclein accumulation is seen in neurons. It's also seen in oligodendrocytes. On the top left, when the alpha-synuclein is in its normal state, it's often called a monomer. And then it starts to aggregate into larger little chains, and these are called oligomers, and then large threads that fold into very complex shapes that we'll be talking about more today, and these are called fibrils. Once this vicious cycle is set up, inflammation on the right side is induced, and I've told you some key players there, the star-shaped astrocytes and the immune cell of the brain, the microglial cell. So I said a minute ago that the alpha-synuclein starts to fold into distinct shapes. Sometimes these are called strains, and you'll hear that term as well. And one of the great challenges of developing a treatment for multiple system atrophy is that all patients with MSA are not created equal. You know that yourselves, and a one-size-fits-all strategy may not be appropriate. why is this disease so diverse? In a nutshell, we think it relates to environmental effects, the fact that the alpha-synuclein can fold into very different shapes, and also in terms of the genetics. Complex genetics could play a role, not the sort of genetics that typically runs generation to generation in a family, but complex genetic factors that in conjunction with themselves and also with these environmental exposures lead to differences among different patients. And these strains are depicted down here, and it is thought that alpha-synuclein misfolds into different shapes, a different set of shapes for Parkinson's, dementia with Lewy bodies, and MSA. This is an example of this, and showing the different types of shapes that synuclein may fold into. This is viewed through an electron microscope on the top left. And in a landmark paper by Claudio Soto, Shanavaz, and colleagues in Nature in 2020, Dr. Soto's group, using a method that he had actually originally invented for prions, demonstrated something very important to the field. And that is, if you took brain tissue or spinal fluid from patients with Parkinson's disease or MSA, and you amplified the material, the way in which that material amplified that related to the different shapes into which it was folded could actually be distinguished. This is a key point. Many of you will know that early on in the disease, it's hard to distinguish, for example, the Parkinsonian type of multiple system atrophy from Parkinson's disease. But this was an early paper that showed us there could be something about synuclein itself. And I won't describe in detail what this graph shows, but it shows the accumulation over time in a particular laboratory assay of synuclein's aggregation. And it aggregates in different patterns in MSA, which you can see in the blue, compared to Parkinson's disease, which you can see in the red. So rather than waiting for a year or two to figure out the clinical characteristics of the patient, this paper gave us hope. It certainly is not a fully solved problem at all as yet, but it gave us considerable hope that we were on our way to specific tests in the clinic that could get patients into clinical trials and access to therapies earlier. Okay, so this is another paper in the same vein. This was published in Nature. And what you can see here are little brown dots on the top right. This is just a zoomed out view of some brain tissue from patients who had different cases of multiple system atrophy. And then you can see that there are some electron microscopy images from each of these cases showing the shapes, and then some very detailed images of how these shapes fold. And it's landmark papers like these. This was from the group of Michel Gueder that started to reveal that not only was alpha-synuclein acting differently in the lab in these aggregation assays, but when you actually visualized it, you could actually see that the shape of the synuclein protein was different in MSA compared to Parkinson's disease. And this was some of the beautiful images from that paper where they describe two very different types of shapes that they could find in MSA patients. So why is all of this important? Well, the first thing is alpha-synuclein could be a very important aspect, not just of diagnosing MSA, but actually driving the disease. We don't know that for sure. And it could be that MSA is a very complex disease and alpha-synuclein turns out not to be the most important driver of the disease. But we know for sure that there are patients who get severe forms of Parkinson's disease who have mutations in alpha-synuclein. So it's a reasonably good hunch that alpha-synuclein is likely to be a very important aspect of either the cause or the progression of multiple system atrophy. So it's important to understand the way in which it is involved in the disease. So that's point one. Point two is if it's involved in the disease, it's a potential drug target. So knowing about alpha-synuclein is important. And in terms of this particular talk, many of you will have heard that there are a number of alpha-synuclein related therapies in clinical trial, and Dr. Rodriguez is going to talk about those in a minute. It is therefore very connected to the ability to diagnose the synuclein problem in the first place. And that's why a biomarker, understanding this concept of strains is quite important, and will give you all, I hope, the backdrop and the logic for why drug companies are really focused right now on this protein. And then point three really plays into a number of other aspects that are related, but a little distinct. If we can detect the different shapes of alpha-synuclein, we might get an early precise diagnosis, be able to track disease progression and responses to drugs, and match the right alpha-synuclein drug to the right patient. That's a critical point. All right, so I'm going to hand it over now for a few minutes to Dr. Rodriguez, who's going to update you a little bit on synuclein-directed therapies. All right, Diego.
Thanks, Vik. So we're going to start by looking at one review published by Dr. Wenning's group last year, where they compiled the different strategies and the different clinical trials that we're actively recruiting at the time. So we'll start by saying that this MSA is tackled, or from a drug development perspective, it's tackled in different ways. So one first step, as you can see in the figure, is trying to genetically modify the way synuclein aggregates. So that's where we have some potential gene therapies, ASOs. We can then try to prevent synuclein from misfolding and aggregating. That's another alternative. Further in the chain, we can prevent synuclein that is already misfolded. We can start clearing those aggregates by, for instance, having some kind of immune-directed therapy that recognizes misfolded synuclein and helps the body get rid of it. We can also prevent aggregation of synuclein so that monomer of alpha-synuclein will not become an oligomer or a fibril. And the last step of the alternative is have a medication that helps us degrade the fibrils of synuclein. So those are the different stages that we're trying to tackle. And we're now going to look at what trials we're recruiting last year. And so as you can see, there's different strategies with different medications that we're recruiting. And I think the key point from this chart here is that most of these trials, except one, were all in phase 1, which means that the primary outcome was really safety and tolerability. Are these drugs safe for healthy controls and maybe some PD patients or some patients with multiple system atrophy? But in general, we were not looking at the time to whether the drugs were working or not, but rather whether they were safe. And now fast forward to today, and this is based mostly on the updates discussed during the Movement Disorders Society conference this year. We have some of these medications and new ones too that have moved into phase 2 randomized control trials. So this is the next step in the ladder for drug development, where we know that the drug is safe. We know that it can be tolerated, that the adverse effects are tolerated. Now we're trying to see if in a small population of patients with MSA, is there an effect that is positive and that we can potentially replicate on a larger sample. Of course, we're still monitoring for adverse effects, but the idea of the trial is now looking to see if the drugs actually work in the population. And that's a big step. We don't know how much of this or how many of these drugs are actually going to work, but at least we've done a big step from moving into phase 2 trials. And now we'll go into biomarkers.
Great. So I'll mention a couple of things. Thank you. We'll move through this fairly quickly now in the interest of time. But we've talked about biomarkers as key for early diagnosis, response to disease progression, and matching the right drug to the right patient. And so the general point here is that we can get biomarkers from any really any part of the body. We can use brain imaging, spinal fluid. We can look at gut microbes, the skin, the blood, which we'll talk about more in a minute, and also get brushings from the nasal cavity to really get access to the nervous system that way. And so in terms of synuclein, you'll see that alpha-synuclein appears here and here. We can detect alpha-synuclein in the skin. We can detect it in the spinal fluid. I told you about the paper from Dr. Soto and colleagues. Just keep in mind, we're not going to talk a lot about at the skin today, but there's going to be an upcoming webinar completely devoted to diagnosing MSA and related diseases through skin biopsies. And so stay tuned for that. And today we're going to be talking about alpha-synuclein in the blood. And really, Dr. Rodriguez will take you through fairly quickly through a paper, but the take-home message here is that it's hard to get CSF. It's a little easier to get skin, but it's very easy to get blood. And so if we could develop a biomarker in blood, that would be perfect for early diagnosis and treatment response monitoring in MSA. So without more ado, over to Dr. Rodriguez again to take us quickly through this paper so that we get the take-home message for you.
Okay, so the first thing we're going to look is at the cohort. So this is a serum assay, seeding amplification assay, which some of you might be familiar with in CSF, but this is done in blood for different synucleinopathies. And so in the next slide, yes, we're going to look at the different cohorts they used. I'm going to focus on the MSA one. It was 39 patients. So it's not a huge sample, but for MSA, it's definitely it represents a good chunk of the population, and also it was validated in a secondary cohort. We're just going to focus on this first one. And so what we see in terms of the results, basically, of this assay is that it was able to successfully detect synuclein in the serum of the participants with MSA with about a 65% of positive results. You're going to see that for Parkinson's disease or for dementia with Lewy bodies, which are other synucleinopathies, the rate of success was higher, so around 95%, which is also consistent with the data we have seen from CSF and other sources of these biomarkers and this assay. For some reason, it's harder to detect it in MSA. But even then, to be able to correctly identify synuclein in 65% of the participants from serum samples is still remarkable.
So this is a great point, and just something to emphasize that even in 2/3 of the patients, it wasn't always positive. 1/3 of those it was a false negative, but for 2/3 of those patients, it could get it right, and that means those 2/3 of patients could be diagnosed more precisely than through our current methods, and those are the sorts of patients that would benefit. Now, these tests get better and better over time, and so this was a remarkable first paper that showed that even synuclein, even though it's a brain protein, even looking at it in the blood could be very beneficial. So I'll advance.
We're going to look how to get synuclein from the blood, because when we do with this type of assay from CSF, for instance, synuclein is abundant. We don't need to do much more to that CSF to really extract the protein. But in serum, in blood, there's a lot of other proteins that we're not really interested in. We really want to isolate synuclein. And so what they did in the first part of this experimental design was to use this magnetic beads that you can see on the tube, the Dyna beads, that are coated with an antibody against synuclein. So basically, these beads are going to capture synuclein and isolate synuclein from all the other proteins in the blood. So you can see how when the serum is inserted into the tube, synuclein or alpha-synuclein seeds, represented at that large figure on pink, is going to bind to the antibody-coated beads. And it's important to remember, these are magnetic beads. So once they have captured synuclein, the next step is going to be isolating synuclein from the rest of the other non-antibody binding proteins in serum. So since the beads are magnetic, we can use that property to keep them attached to the side of the tube and then collect the remainder of the fluid, do multiple washes to make sure that we don't have anything else other than the beads, the antibody, and synuclein, and then we will elute and neutralize the solution to separate the beads and the antibodies from synuclein. Now, once that's done, we have a solution that has serum synuclein that we can then process through a seeding assay, which basically pairs each strain of synuclein from the patient with recombinant synuclein that we make in the lab. And we basically look at how they aggregate over time. And there's a marker, thioflavin T, that is going to give us fluorescence. So the more they aggregate with thioflavin T, the more fluorescence we're going to see. And we get up building these curves of THT fluorescence over time. So I'll walk you through how the result looks like and what does this mean for MSA and PD diagnostics.
That's great. So I'm just to recap, there's an antibody here capturing synuclein, and just as Dr. Rodriguez says, there's a chemical that binds that particular shape that synuclein folds into, an amyloid shape. And so this chemical is thioflavin, and it's got this beautiful property of glowing in a color that we can detect, and that's what you're seeing here. So as alpha synuclein aggregates, it's been captured from the blood now in a purified form because of that antibody. We can watch it aggregate, and it likes to aggregate. So all we need to do in the lab is give it artificial synuclein, and then the synuclein from the blood will latch onto it and make more of itself. And that's what that synuclein monomer that Dr. Rodriguez was mentioning. Okay.
And so this is how the result looks like. So there's a couple things. Dr. Khurana mentioned how based on the shape of the curve, we can start to differentiate between Lewy body diseases and MSA. But more than that, what really this assay helps us do is to solve the question whether there are synuclein strains that are aggregating and templating over time. Is basically, are these synuclein proteins that we are capturing able to aggregate with normal synuclein? And there's a couple of metrics we can look at, which is how long it takes to aggregate, how much fluorescence we can measure. But the general concept is, do we see or not synuclein that can aggregate over time? And as you can see, this resulted to be a very sensitive, very specific assay for PD, which gives us a high diagnostic yield for Parkinson's disease. And although not as high for MSA, is still extremely useful. And I want to emphasize that is as good, or seems to be as good as the one in CSF, which is key because serum is much more accessible than CSF. Now, the second important part of this study is that previous studies in CSF, such as the paper from Soto and colleagues that Dr. Khurana was discussing before, have showed that there are different structures for these strains for synuclein between multiple system atrophy and Lewy body disease. And in this paper, these authors identified the same differences in the structures that we see in CSF. They identified those differences in blood too, which means that we are actually capturing those same strains with the same structure from the CSF into the blood. And the way they did this is through a technique called transmission electron microscopy. So this is basically a very fancy way of imaging small structures. And you can see by the presentation of each figure that MSA fibrils are not paired. They can be twisted, but they're single fibrils that are wider than Lewy body disease fibrils. Lewy body disease fibrils are thinner. They are usually bundled or paired, and there's a significant difference between the width of the fibers. So this also helps us think that the structure is actually different, and this is how it looks in the microscope. So you can see PD kind of two different fibrils with a space in between. And for MSA, you just see a large fibril that is either twisted or straight, but it's definitely not paired with any other.
Mhm. Great.
And the last portion that we're going to discuss today is that not only we find a different structure for those fibrils, but it appears like there's also a different structure for the inclusions, the aggregates of synuclein within cells. So this is a experiment where they used a very widely studied cell model for synuclein disease that has a mutated synuclein that normally does not aggregate, but is tagged with this green fluorescent dye that will make it look how you see in the image. So it's going to give us this green dye when it aggregates. And to those cells, they introduced the material that they amplified from each of the patients. And the key finding was that there are three different types of inclusions. This FL inclusion is kind of a fibrous inclusion. The DC inclusion has a dense core that you can see that very bright region. And the PC inclusion has a pale core, kind of a more dark region. And it seems like this each type of inclusion correlates with a different disease. So when they looked across the samples, multiple system atrophy appears to have both dense core inclusions and pale core inclusions, while these ones are not seen at all in Parkinson's disease and in dementia with Lewy bodies, in Parkinson's disease dementia. Uh, the pale core inclusions are also present, but the dense core inclusions are not as common as they are in MSA. So this opens the door to have cellular-based assays that could also help in the diagnosis of these conditions, since you have different structures.
Yeah, so that's terrific. And I think that's the last slide we have on this particular paper. And again, all in the same theme, whether it's putting these fibers into cells or measuring them, their fluorescence readout, like that we just showed you, that thioflavin molecule. The idea is that these different diseases are acting differently in the lab, and we could detect them from a blood-based test, which would be really a game changer. So we're not there yet. This is not a clinical test. I think that's really important to stress. This is research, but it was a significant enough advance for us to really feel that we should highlight why it was important, especially in the context of the many different synuclein strategies that are in clinical trial or heading to the clinic. Now, this kind of research has been tremendously enabling for us as well. And so we wanted to, very briefly in the last couple of minutes, just tell you a little bit about what we're doing in our center, and equally centers all around the country that are researching MSA have been really transformed by these kinds of discoveries. So I'll hand it over to Diego in a second, but essentially at the Brigham, we are looking at developing innovative therapies for MSA, and we're tracking our patients over time. So at last year's conference, I discussed this whole paradigm of taking a patient into an observational phase, giving them the drug and seeing how they responded, then giving a placebo, and then giving them the treatment again. So it's a different type of paradigm. But in order to do this well, we need really good biomarkers. And that's where the paper we just discussed really is important. And so, Diego, can you tell us a little bit about the types of things that we and others around the country are collecting in our clinical trials units?
Yes, so very briefly, I think this different groups are doing a subset of this test. We have incorporated into that tracking before we test the medication. We really want to make sure that we have the diagnosis correctly. And so the first group is actually, I'm going to start with the yellow area, is those biomarkers that are promising for identifying synuclein. So blood, CSF, synuclein from skin, and of course iPSC generation, which is a big part of the focus in our lab. And then on blue,
just to chime in, iPSCs are the induced pluripotent stem cells I've talked to you in the past, and I'll show you a slide of that right at the end.
Yes. In blue, we have those clinical metrics, both automated measurements through wearable devices or computer tasks that help us track uh, basically movement abnormalities and quantify disease progression, also using eye movements. So that's the video oculography you see there. And of course, the clinical scales we always take, right? And then on the green portion of the graph, we have the imaging part. So we've been doing volumetric 3D brain MRI, and this is now incorporated in a lot of industry-sponsored clinical trials as a marker for MSA. But we've also expanded to look for neuroinflammation through PET scans and synuclein into the brain. So if we can see synuclein in the brain, that would also be something we can measure to see disease progression and response to medications.
Okay, so speaking of that, there are a couple of initiatives going on, tremendously exciting, that we may be on the cusp of visualizing synuclein in the brain. There have been a number of different papers here. In our particular clinic, we've been using a tracer from a company in Switzerland called AC Immune. You'll recall from last year that they press released that they had the first images of alpha-synuclein in the brain of patients in MSA. So that's exciting. We're also developing our own tracers for alpha-synuclein, and that's work sponsored by the Michael J. Fox Foundation. So Diego, do you want to show us, take us through this image here?
So in this image, you can see two controls in two MSA cases. And basically what we're looking for in the PET scan is for that SUVR is the way we call the signal. When it turns red, it indicates that the uptake of the tracer is high in that region compared to another region of the brain. And so you can see how the control versus the MSA-C case, you see a red signal around the brainstem that actually is what we call the middle cerebellar peduncles, which is a key region of atrophy in MSA and is actually part of the diagnostic criteria. So it seems that the tracer is able to detect some of the this nuclein that we have in the middle cerebellar peduncles, and it gives us a good signal that we're trying to replicate actually now with the larger cohort.
Yeah, that's terrific. So just to be clear, this is the AC Immune tracer. This is a PET scan. You can see the eyes of the patient, the nose of the patient's on top. You can see the cerebellum at the back of the brain, what Dr. Rodriguez is referring to as the brainstem here, the pons right here. And this was this very critical region that he mentioned, the middle cerebellar peduncle right here, that's glowing red in MSA, but not in these unaffected subjects. So it's exciting times where we may be able to visualize alpha-synuclein in the brain, as well as detect it in the blood. So this is an example. We talked about these iPSC models. And so the paper that we talked about today took a cell line that is like a cancer cell line type model, but it glows when synuclein aggregates. So as Dr. Rodriguez said, a very, very nice way of looking at the synuclein that was being captured by the blood. But what we'd really like to do is capture the synuclein in a model made from the patient themselves, so that we really had a model of that patient's disease for personalized medicine. So we're trying to prepare for that, and we wouldn't be able to do that with the kinds of advances in biomarkers that we talked about today. So the basic idea is that we can take spinal fluid or blood now, and nasal brushings, sometimes biopsying the gut for some patients in particular circumstances, or after death, looking at the postmortem brain, and we can visualize synuclein in those tissues. So this is visualizing synuclein in brain, skin, or the gut. Then we can amplify it just in the way the paper we described today discussed. We can take the abnormal synuclein from the patient's tissue and then combine it with an artificial synuclein in the lab. And when the abnormal meets the normal, it causes a templating reaction. So you get much more of the abnormal synuclein being misfolded in the reaction. And then you can visualize it with these fluorescent chemicals. We talked about thioflavin T and the pattern of MSA and Parkinson's disease being different. This work is being led by Dr. Kuskina in our lab. And once we have a large amount of that material from the patient, we can introduce that back into neurons that we've generated from stem cells from the patient. So these iPSC models, and you can see here MSA aggregating in neurons that we've made from a patient, MSA synuclein versus Parkinson's disease synuclein, looking different. And this work has been led by Doctors Ndesaba and Lam in our group, and this paper is on the cusp of being published and is available in a preprint form. So we hope we've introduced, the importance of biomarkers for MSA, why the paper from Nature Medicine from the Hattori group we thought was worth highlighting in the context of everything else that's going on, and how we continue to move as a field to get better and better at diagnosing and hopefully treating and changing the outcome of this disease. Every year, the progress is really astonishing and fills us with optimism. We're nowhere near there yet. It's important to be realistic, but there's tremendous progress and interest. And we hope we conveyed some of that to you today. This is always one of my favorite talks to give. This year, I'm particularly excited that I got to give it with Dr. Rodriguez, who's just an amazing colleague to have in our group. And as always, our greatest shout of thanks to you, our patients, and the caregivers. Thank you so much, and see you next year.